everybody. It's my pleasure to welcome and introduce Kumaraswamy Guruswamy, who was in NCL, who worked in NCL, and just a few days ago has joined IIT Bombay in the Chemical Engineering Department. I know Guru since I joined IISER Pune in 2009, and he has been quite an intriguing character for me. Why? He's quite unlike Ravan in that he has only one head, but he manages to wear many hats simultaneously. Let me explain what I mean by that, and let me talk about what he has been doing. So he did his BTech from IIT Bombay in 1994, chemical engineering, and after that he moved to Caltech to do his MTech and PhD in the chemical engineering department of Caltech, which he finished in 2000, year 2000. Then he was in Max Planck Gulm in the department, in the Max Planck for colloids and interfaces, where he did a postdoc for 1 year and joined NCL in 2001. So he is a chemical engineering by training. He publishes mostly in ACS, American Chemical Society journals, but seems to be most appreciated by physicists. What do I mean by that? He is being elected the APS, American Physical Society Fellow, in 2016. Basically, his citation reads like, for opening new routes to the templated polymeric structures using mesophysics and crystallization and elucidating their physics. He is also represents Asia in the IUPAP, the International Union for Pure and Applied Physics, and he is in the working group for soft matter physics. He has been working with many of us in the physics department, at least two, with the faculty who are in chemistry in IISER Pune. So when he's talking to me, he talks like a physicist. I guess when he talks with the chemistry faculty for collaborations, he must be doing a very good job there as well. But then once in a while, he puts on his chemical engineering hat and does consultancy and does projects with many multinationals and has designed many interesting products, starting from the physics and chemistry, which he does with us. And I think he will be also talking of such example where he will talk about the physics of the problem, the chemistry of the problem, and will talk about a certain application which has helped many or is going to help many. And you shall hear about it from Guru himself. He has worked with many companies like Reliance, Indian Oil Corporation, GE, Procter Gamble, DuPont, and earned quite a few crores for NCL as part of his consultancy. He's an experimentalist. He introduces himself as an experimentalist, but I know that at least one of his PhD students had done his PhD using theoretical techniques and is currently a faculty in IIT Guwahati. Last but not the least, he's part of an extremely active outreach program where he teaches school students. So every Sunday, he organizes camps where he talks about science to school children. That's quite something with this entire range. And without much ado, let me invite Guru to give his colloquium.
Thanks, Apratim. Yeah. So I'm delighted to be here. It's always fun to be here at IISER Pune, and I'm very happy to talk to you about some things that we've been doing in our group. I guess wetting was probably not the right topic to have picked today, but you know, be that as it, I'm glad that there is an audience, and I hope I hope people will come in as I continue to speak. So I'm going to talk to you about some fun we've had with lipids, and I'll tell you, I'll tell you about what lipids are, what assemblies are, and what we've been doing with trying to understand bending and wetting of these systems. So as Apratim said, for the last 4 days, I've been faculty in the chemical engineering department at IIT Bombay. But before that, I was at the National Chemical Laboratory right next door. So all the work that I'm going to talk about was done primarily in Pune. And you just see there's a pointer here. I list my affiliation as IIT Bombay, but there's a little CSIR symbol at the top there just to indicate that all of this work was done really in Pune at NCL, right next door to IISER. So before I start, I want to give credit where it's due. There's clearly some very nice work that was done that I'd like to talk about. Most of this was done by a very talented PhD student, Manoj, who had a lot of fun doing this work and did some very nice work, as a consequence of which he received the Best Thesis Award, the Kirti Sangoram Award, for the work that he did as part of his thesis. And then, like many other smart people do, he's moved to biology. He's very interested in biochemistry and biophysics, and he's currently a postdoc at the NCBS in Bangalore. Several other people have also helped with this work. Mayuresh and Arun Banpurkar at the University of Pune were especially useful when we came to having discussions about wetting. Some of the simulations, I'm not going to spend much time talking about that, was done by the group of Sudeep Roy. Chandan was the student who did this work. And one of the fun things about being at NCL is just the access you have to chemists who are willing to make very interesting molecules for you. So we're very grateful to Nagnath, who worked with Ashutosh Ambade for some of the work that they did in making some very nice molecules that helped us prove a point that we were trying to make. Like a lot of other fun, interesting science that is supported in the country, this work was supported for 3 years by the DST. And we had a lot of fun doing this work. And we didn't, I just want to sort of make the point here that when we started doing this work, we set out to try and understand a few things about lipids and lipid assemblies. We did not set out to develop a technology, but something that magical sort of happened along the way is that a very nice technology got developed. And at the end of it, this has recently been licensed to ATGC pheromones. The point I'm trying to make is there's a really important role that serendipity and just mucking around with experimental systems has in also things like technology development. So sometimes technology emerges, and if you don't support the science, the technology doesn't happen. Incidentally, ATGC is just the upfront payment for the technology without royalties, and any of that is more than what DST paid for 3 years of support for this work. So it's just a point I wanted to make in terms of supporting basic science. Okay, so let me tell you a short story about things that we've done. So this is mostly about lipids, right? I'm sort of getting to the age when I worry a lot about lipids, things like fatty acids and triglycerides. Those are the things that I look for in the pathology reports, and not with some sense of trepidation when I do that. But lipids are how the body stores energy, right? And so these are largely hydrophobic molecules, things like cholesterol, fatty acids, triglycerides, things like phospholipids, and so on. And so while they're mostly fairly hydrophobic molecules, they're also, some of them are amphiphilic in the sense that they have parts that are hydrophobic, that don't like water, that are connected to parts, this is the head group, that like water, right? And so there is this schizophrenic character that these molecules have, and as a consequence of which, they end up assembling into a wide variety of structures, right? And I give you an example here of a structure. So this is a cartoon representation where the green is the hydrophilic head group and the yellow is the tail. So this is a two-tail lipid, and on the inside are these hydrophobic molecules that the tail likes, and on the outside is water, right? So that's an example of lipid self-assembly. And the reason we care about lipid self-assembly is kind of obvious, because lipid self-assembly is extremely important to life, right? And I don't know if Sudha's in the audience here today, but things that we worry about a lot are this kind of a skin that goes onto the cells, right? So this is a bilayer, right? So you've got these yellow tails here. In this case, you've got red water-loving head groups here, and they form this bilayer structure, and all our cells are covered by this bilayer. So this is the cell wall that goes around it, and there's an enormous amount of interest in understanding chemical heterogeneity, structural heterogeneity, mechanical heterogeneity in these systems, some of which is driven by a variety of molecules that are also embedded in these bilayer systems. So compartmentalization to form these cellular structures is essential to life, and is essential to a wide variety of things that make life healthy, right? And so that's a big reason why people are interested in understanding assembly in these kinds of lipid structures. Now, these lipid bilayers, the ones that I showed you, they look like a flat sheet, but they're also characterized by curvature in many instances, right? And curvature is sufficiently important in biology that nature has actually developed mechanisms to sense this curvature. So I'll give you an example from the literature of a phospholipase. So this is an enzyme that crawls along the bilayer of the cell, and it's a phospholipase, so it goes in and chews up some of these lipids. And what's been observed is that the activity of this enzyme is a function of the curvature of the membrane that it crawls on, right? So if you have a more curved structure, then it ends up being more active and degrading some of those lipids, right? And so there's a variety of biological reasons why curvature is important in these structures. It tunes activity, and therefore it tunes, in a sense, health in living organisms. There's also technological implications. So what I'm showing you here are complexes. These are synthetic complexes made using liposomes, which are lipid assemblies, with DNA, right? And what you see here is DNA organized to form these different kinds of structures which are characterized by lipid assemblies with different curvature, right? And these are synthetically made. This is very nice work from the group of Safina and Santa Barbara, where he showed that if you were interested in things like gene therapy, if you wanted to deliver some of this DNA to cells and then to the nucleus, then the efficacy by which you can take these constructs and have them transfect the DNA depends on the curvature of these assemblies that you see in these systems. Therefore, curvature has technological implications, important in biology, and that's something that we should care about a lot. How do you understand why membranes curve? How do you control it? That's something which is really important to us. So let's go back and sort of take a very sort of a bird's eye view of self-assembly. So here's again our cartoon schematic with the head group attached to a hydrophobic tail. So this likes water, this doesn't like water, and so they're schizophrenic and they're attached, right? So these are covalently bonded, so there's no way they can escape from each other, right? So oil and water will demix if you put them together, but in this case, when you've chemically bonded them, they really don't have that option of doing that, right? And so lipid assemblies really comprise of these schizophrenic molecules that come together and then organize in a way so that everybody is more or less happy, right? And so if you think about what guides this assembly, it's really things like the interactions between the head and the head and the tail and the tail, and so on and so forth, right? So there's a myriad of assemblies. The head groups often end up being charged, and so there's strong repulsive interactions that try and force the head groups away, while on the other hand, there's attraction between the lipid tails. Again, there's attraction and sometimes there's repulsion because you want to have enough wiggle room so you can't pack them too tightly beyond a certain point, right? And so there is this complicated balance between the interactions in these systems that really tells you what kind of structures form. So this is again taken from a review paper which shows the lateral pressure in these systems. There's all sorts of very complicated interactions, entropy, enthalpic interactions that give rise to the lateral pressure that you see as you go down this lipid bilayers, right? So when you start thinking about how these things curve, here's a really beautiful textbook picture that's really comes from the work of Jacob Israelishvili, again, who's from Santa Barbara, unfortunately passed away recently. And what he said is that if you have this structure and you have this self-assembled structure that comes from all these interactions, let's not worry about that complexity, right? So at the first instance, all you want to do is to take this system and think of it as purely a simple geometric packing argument, right? So I take this head group with a tail, and what I can then do is to come up with a parameter which is called the packing parameter. It's just a geometric ratio of the area of this tail to the average area that's given by the head. Right. Now this area is determined by all these interactions, but this simple geometric parameter then allows you to say what kind of structures form in these systems, right? So you start off with systems which are micellar, so this is like soap that we use to wash detergents and so on. So you've got a head group that's large and forms these roughly spherical micellar systems. And as you ramp up the size of the tail, you go from a spherical structure to a cylindrical structure that can organize to form hexagonal packing, to structures that have local cubic symmetry. These are the bilayers, and then go all the way to form inverse phases, right? And the inverse phases are where you have a lot more of the hydrophobic groups compared to the hydrophilic groups. And that's one of the things that we're going to be interested in today, is to talk about some of these inverse structures in these systems, right? So very beautiful, very elegant, very simple picture that Israeli gave us many years ago. And this has really stood the test of time in the sense that, this is the one thing that we start from when we start thinking about how lipids assemble, how surfactants assemble in these systems, right? This very simple picture that you have to take the ratio of the head to the tail. If the head's too large, then it sort of forms a sphere. And if the tail is too large, then forms a sphere in the opposite direction. And it goes through the entire series of geometric structures that you have as you go varying the relative size of the head and the tail, right? So that's sort of a preamble to what I'm going to tell you about, is some work that we started doing some years ago using a monoglyceride. So this is glycerol, and then I take a monoester using oleic acid. It's a very simple molecule. It's a fatty acid. It's a commercially available material. You can get it from sunflower oil, and it's sold in ton quantity. So you can just buy this very readily. It's non-toxic, it's safe, it's food derived. In fact, it's used as a food emulsifier in many applications. And we thought this would be a nice, simple model system, easily available, that will allow us then to study how lipids assemble, what kind of structures they form, how they bend, how they curve, all that kind of fun stuff, right? What's interesting is that there are also pharmaceutical grades of this material, and that's used for controlled delivery of drugs, a variety of active molecules, and so on. And this is a simple molecule, but look at the richness of the phase behavior when you take this one molecule and add it to water, right? So what you have here is a composition of this molecule plus water, and so this is no water, and as you add more water, you go along this axis, and this is temperature on this axis. And what you can see is that as you add in more water, this is a fairly hydrophobic molecule, it starts forming lamellar phases, and then beyond that, all kinds of complicated interconnected phases in these structures, right? That's fascinating, right? Because it then tells you that with this very simple system, you can now access a wide variety of organization in these structures. And what we really set out to do is to try and see if you can play around with this. Now, as you go across, what really changes is you change the amount of water in the system, it changes the relative size of the head and the tail, you change the curvature, and that's really what you're seeing as a change in the structure that you have in these systems. So a question that we asked is that, can you then try and do something interesting with these materials? So yeah, sure. So you get all these different kinds of features. How do you characterize them? Just one slide to tell you that we use a tool called small angle X-ray scattering. It's a very powerful tool, but it's sort of a niche technique that's used to characterize these materials. Niche largely because there's not that many people who are familiar with analysis using this technique. And so this is the phase diagram. I'm going to focus on one part of it, and what you notice here is what's called the PN3M phase. The PN3M denotes a local symmetry in the structure, and is characterized by a series of peaks that you get from small angle X-ray scattering data. And you get these peaks in a certain ratio, and that's what allows you to then characterize the kind of structures you have. So depending on whether you have an IA3D, an FT3M, hexagonal, and so on, you get a different series of peaks. And that's how you characterize the system that you get. You characterize the geometry you have in the system. Now, I'm going to come back to this in just a minute, but notice that there's a PN3M phase, but if you add more water to it, it doesn't sort of dissolve, but it coexists with excess water. This stuff is so hydrophobic that you can't dissolve it to form these lower concentration structures. You go beyond a certain amount of water, then the PN3M phase exists, but it just exists with excess water. So it's sort of like this really annoying thing when you open this dabba of dahi and you have this water floating on top. That's not necessarily a nice thing, but what it tells you is that the assembled structure in the curd is able to exist with the excess water in that system. And these are equilibrium phase diagrams that you see here. That's not the case in dahi, but this is an equilibrium phase diagram. Okay. Now, why is this interesting? This is interesting because, as Apratim mentioned, I'm also a chemical engineer, right? And so I worry about how I'm going to process this stuff, right? So if I take just this small molecule that flows, it's an oil that flows and I mix it with water, I end up with a material which is remarkably stiff, right? This is amazing, right? Just the way in which it folds to form these structures makes this system have a viscosity which is 1 lakh Pascal second, which is 10 million times that of water, right? And so you take this, and so you have my students sort of pushing down on it with a spatula. This stuff is really like a rock, right? So imagine if I take a reactor and put this stuff in there, it's going to be virtually impossible for me to stir this, right? So it's very hard to work with. So even if I do something interesting with it, I can play around with the curvature. How do I then practically do anything with it if I want to sort of make a product out of that, right? And so one of the things that we found that was interesting is that because you have the PN3M phase that exists in equilibrium with excess water, I can take that material, chop it up. This is an electron micrograph, which shows that if I chop it up into, this is about 200 nanometer little chunks, the internal structure that I have in the PN3M phase is preserved, but now it's surrounded by water. So I have a material which essentially flows like water. It's very easy to process, and I have to put a little bit of polymer around that and so on, and you get this very low viscosity dispersion that I can then play with, right? This is data that we got in collaboration with Jayesh Bellari's group at IIT Bombay, who do absolutely beautiful electron microscopy in these systems. We were not really the first to come up with these systems. This is the phase behavior of these systems have been understood for quite some time, but we started doing something very interesting with this, and I'll tell you more about that in a little bit. Right? So I'm interested in this PN3M phase, and this PN3M phase is what's called a bicontinuous structure. So you've got channels of water and channels of oily hydrophobic stuff that are interconnected and that sort of percolate through space, which means if I want to take in this material, right, and I want to take this material and I want to put something which is a water-soluble drug in the aqueous channels, it just leaks right out, right? And that's not very useful to me, and therefore what we started thinking about, and that was really the genesis of this whole project, is to start thinking about, is there a way by which I can get it to curve such that I have a little drop of water surrounded by oil. Then if I put a water-soluble molecule in there, it doesn't leach out very, very easily. So I'm not going to tell you about the work that we did in terms of release from these systems. I'm going to tell you about how we started thinking about how to get these things to curve, because I have materials here that are locally sort of like flat sheets. They're bent around to form these interconnected structures. But how do I get them to bend even more so that I can get a little drop of water that it holds, right? And the reason I want to get this discontinuous phase is that I want to try and encapsulate some drugs in there. And I want to do this at equilibrium so that if I make a pharmaceutical formulation, I put it on the shelf, it travels around, it goes to Nagpur, it comes back here, you don't change phase, because that would be disastrous if you're doing it with a pharmaceutical molecule. So that was really the origin for why we started looking at this. And then when you start thinking about it, you realize that this is actually a pretty hard problem, right? Because if you have a regular oil and water phase, a little drop of oil surrounded by the surfactant, that's how we wash our clothes, that's how we wash our bodies, that is standard detergency. Because if I take this detergent, then I can take a lot of the dirt that we have is oily, it just gets washed off, it goes in there, and I'm surrounded by water, so I can get this nice curved regular structure. How do I get an inverse structure? Now the reason that's hard is because now what's around you is the hydrophobic oily part. And so if I want to take this and I want to get it to curve the other way, then I need to be able to fill more of that space, the space on the outside, with this oily part. But I'm not floating around in oil. All I have is this lipid molecule that I have, and therefore for it to fill space, right? Nature doesn't like to have empty bits of space lying around when you make anything. So if you want to fill space, then either I have to put some oil in there to pack that extra space, or I have to have these lipid tails stretch. Right? These are C18, C20 chains. They don't really want to stretch, right? And so that becomes a really, really difficult problem to solve. To get inverse phases where I have little drops of water surrounded by oil, then turns out to be a really hard problem to get to, right? So if you think about it, if I have a hexagonal phase, I have cylinders of these lipids, then I can do a very simple geometric calculation, and this little bit that's excluded, right? If I just take cylinders and I stack them and I look at the volume that's excluded, that's about 9%. It's not a lot, and so typically it's much easier to get these things to form these kinds of hexagonal phases, if I take a cubic phase, right, I take balls, I take equal sized balls and I stack them, the packing fraction is 74%. We know that from, this is the standard maximum packing fraction that we get in these systems, which means 26% of this is interface. I need to fill it with something, right? That's really hard, right? So if I want to form a little ball of water surrounded by oil, I've got to then figure out a way by which I fill out that 26% of space with something like oil, right? And so typically the way people do it is to put oil in these systems. If you take, let's say, vitamin E, tocopherol or something, you put it in there, then you can get these kinds of inverse phases in these systems. But we didn't want to do that. So we thought, let's go back and try and understand how this has been done in the past, in other systems. So for example, if you take inspiration from nature, one of the things you see is to try and get lipids to curve, get lipid assemblies to curve. What is often done is you embed it with some other molecule, right? Like a polymer, like a protein, like an enzyme, and sometimes that also controls the way in which these lipid assemblies curve, right? So for example, you can have proteins embedded in these and so on and so forth. So the thing that we started thinking about is, we're starting off this nice simple model system. What about trying to see if we can embed a polymer in these systems? If I put a polymer that wants to go into the system, could I get this to curve? Now, the question then that comes is that what is the effect of polymer shape on the curvature? And this is the part that gets difficult because there's some experiments which are difficult to interpret, but then there's a lot of theory, and depending on which experiment you want to explain, you can get it to curve this way or that way, whichever way you want. Right? So it becomes very hard to then start off trying to guess what's important in these systems. And so this is work that comes from the literature, from the PhD thesis of a group in Cologne. And so we started thinking about this whole idea of shape, right? So again, one of the things that I think we are typically bad at is to think about things in 3D, right? So when you think about a molecule, you write down a structure, you think about it in 2D, but really these molecules have three-dimensional shapes, and so do complicated objects like polymers, right? And so when you want to start thinking about this in 3D, then you want to start thinking about, for example, how do you control the shape of these structures, right? So when we think about a polymer, you think about this long linear object, but that's not the kind of compact objects that you see that are embedded in these bilayer systems. So one of the things that we thought we should try and do is to play around with the shape of this molecule, right? So we have here a linear molecule, and so the little red dots are amine groups that interact with the surface, and then you make match systems. Again, this is where the chemists in NCL came in making these very nice molecules, and you can match these so that you have the exact same number of these functional groups in these systems versus this. So you can make systems with second generation, third generation, fourth generation of these beautiful compact shapes, and then match linear molecules which have the exact same number of molecules in there, right? Beautiful engineering of these molecules. And then when you put it together, we're going to keep this, I'm just going to get straight to the results. This is what happens when you take the monoolene or the glycerol monooleate, the lipid, water, and the fourth generation dendron, and you compare that with the linear equivalent of the fourth generation dendron. What you see is that at very small fractions of this water soluble, with the amine, these are highly water soluble polymers, you can start getting these inverse phases, which are called the FD3M phases. I'll tell you what that is, while you can never get that with the linear molecules, right? So what this tells us is that the shape of these proteins or compact molecules that you put in with this lipid water system is critical in determining the kind of shapes that are the kind of curvature that you get in these systems. So what's the FD3M phase? Let me just sort of quickly show you. It's an amazing phase, right? So remember, typically what you would expect is you would need to put oil into a system to get it to curve the other way around to make these inverse phases. What we're putting in this is a water-soluble polymeric molecule, right? The second thing is, remember this thing we talked about. If I have a hexagonal phase, I need to pack 9% extra volume. If I have a cubic phase, I need to pack 26% extra volume because I have 74% that's filled. What you end up with is a phase where you actually have 29% extra volume, right? And this is this phase where you have not just a single size of these micelles, but you have micelles of two sizes, a big sphere and a small sphere. And so it's called an AB2 phase. You have two small spheres for every big sphere, and it packs into this very beautiful cubic geometry with a little drop of water on the inside, right? 29%. Why would you want to do that? And that's where you can't second guess nature. turns out that this packing actually turns out to be the most optimal in terms of adjusting for the frustration of these lipid chains. And that's something which it would have been impossible to guess or to do a calculation to figure this out. Right? So the chain stretching is actually less than what you would expect for a system where you would have only 26% of the interstitial space. Here you have more interstitial space, and yet you can get this to form. And this kind of phases had been known in the past, but typically you need to put in 40% of oil to do this. Here we're doing this with 1/10 of it, 4% of a hydrophilic molecule, right? And this was completely unprecedented. Now, why do I tell you all this? Because the point I'm trying to get to is that this kind of a result is completely at odds with the textbook picture, right? So the textbook picture tells us you need to look at the size of the head versus the tail, and by putting in this hydrophilic molecule, you're really not changing this as much, right? If you complex the head group with the dendron, you can't get a 3-fold decrease in the head group size that you need to make this change happen. And plus, this is actually an uncharged molecule, so you really don't have charge effects in here. So what this tells us is that, great, this is a textbook model that's worked, it's beautiful, and for the most part it does work, but you shouldn't use that to limit where you think you can go with this, because really there's all kinds of things, all kinds of surprises nature throws at us, which are really outside of what we learned from the textbook. So we did some molecular dynamics simulations in collaboration with my colleague to really show us what happens. So what happens here is that the compact molecule, which is this dendron, can't spread on the surface. It really likes to complex with the surface but can't spread on it, and so it just folds the surface, while the linear molecule likes it and so sort of spreads on the surface rather than having to fold it. So just a quick version to let you know that we actually do understand what's going on in these systems. Now, what I did tell you right at the beginning is that the structure of these assemblies, the curvature, is really determined by a whole host of interactions, and it's very complicated. So really, apart from the geometric packing parameter, there are no rules that tell us that if I want to design a certain structure, what do I need to do? How do I need to go? These experiments of ours really showed us that we have some general rules that allow us to predict this. So here's another molecule. It's again a commercial, cheap, easily available branch molecule. But again, because it's branch, it's compact, and as a consequence of that, you can get all these kinds of FD3M phases. Now, the reason I show this is to tell you that, while I can come here and give a talk, it took Manoj a year to generate this one slide. Right? Each of these data points is data where he's done microscopy, he's done small angle scattering, he's varied temperature, he's varied concentration. So it's an enormous amount of data. It's 1 year's worth of work, but it tells us a beautiful story. Well, it also tells us a story that you need to work really hard to generate the data that goes behind the pretty pictures that you can show. Right? And so this is some very beautiful work that emerged from Manoj's thesis. So what this teaches us, so this is sort of the first part in terms of bending, is that that if you want to generate curvature in these systems, the packing parameter is a good starting point, but there are other ways, and these are probably ways that nature exploits as well. The series of experiments that we did allowed us to come up with predictive rules to design how you can get curvature in these systems. What kind of additives do you need to add? What we saw is that the additive needs to be compact. If it is in itself not a very well-defined shape, it can spread, and it needs to have a strong attractive interaction with the head group of the lipid. And so that's really the first part of the story. So the second part of the story turned out to be more interesting, and this has to do with how these lipid assemblies behave when you go near a surface, right? So you have these lipid assemblies, and remember that I'm going to focus now on these cubosomes that we talked about, these little chunks, 200 nanometer chunks floating in water, low viscosity dispersions. And the reason we got interested in this is again driven by Manoj's fascination for this work. Manoj comes from Himachal, and his family is into farming, right? So they work on apple farms. And one of the things he would keep telling me is the amount of pesticide that one sprays on these farms is just stunning, right? We live in a tropical country, so we are happy living here. We don't have to deal with extremes of hot and cold. But at the same time, this is wonderful for a lot of bugs as well. And so for us to not use pesticide is really, really difficult. I'm going to come and talk to you a little bit about not using pesticide at the end. But one of the big problems is that because of the large volume of pesticide that we use, the pesticide essentially not all of it gets onto the plant. It leaks off the plant, falls into the soil. It contaminates the soil, gets into our water streams, and this is a really big problem. And that's one of the reasons we find pesticide residues in all kinds of things. So turns out that the pesticide problem is actually a fluid mechanics problem, right? And that's again my chemical engineering hat. So here's what happens, right? So if I take a plant surface like a leaf, the leaf is covered by a thin layer of microcrystalline wax, which makes it very hydrophobic. And so if you take a drop of water, put it on a leaf, it will typically bounce off. So here is an example. This is actually a model hydrophobic surface. This is a glass slide which is made hydrophobic. And this is a highly sped up video of a drop that impinges on the surface, right? So it's about a 2 mm drop. And this entire video is about 20 milliseconds long. So the whole time that it takes for the drop to come impinge, and it's spectacular, right? In 3 milliseconds, it spreads out and then comes back and then bounces off the surface, right? And this is similar to what you see when you have a plant leaf. For example, this is a rose leaf just outside my office, and if you have a drop of water, it just slides right off, right? And this is the problem because when you take these pesticides, mix them up with water and you spray them, they just bounce off the leaves, and that's the reason you have a lot of pesticide that gets into food, water, soil, and so on. So one of the things that Manoj was very interested in is to try and understand these very hydrophobic lipids, right? So these hydrophobic lipids that form these PN3M phases that form these cubosomes. If I can encapsulate pesticide in these, and that was the initial thought, then can I get it to stick more efficiently? Because remember, this is a molecule that's fairly hydrophobic. It's sort of delicately held together by this balance of forces. But what happens if I take this close to the surface, right? So here is an example. So let me just play the previous video so that you remember what it looks like. Bounces, just comes right off, and then bounces right off the surface, right? Now here is a drop of cubosomes, 1% cubosomes in there, spreads exactly the same way, but notice it doesn't bounce off at all, right? It just gets stuck to the surface that you see in this material, right? And it stays stuck for a long time. This video is still playing, right? If you do it on the rose leaf, right? Yeah, it's an inclined rose leaf, it sort of flows off, but note that it's not really flowing off, it's stuck. You wait long enough, you can actually see a flow in the opposite direction, right? So there's actually an adverse pressure gradient that's going against gravity that's holding this stuff on the surface, right? So this is absolutely fascinating. And so of course, we went back and tried to quantify it. So what I'm plotting here is the size of this drop. So I start off with this 2 mm drop. It's all normalized by size. I start off with this 2 mm drop, splat, goes onto the surface, spreads out, and then it sort of retracts, right? And what I'm showing you here is data for a drop of water. That's what I showed you in the first slide. And then what happens if I start putting surfactants in it, right? So this is again one of the things that farmers do is if I want to spray a pesticide, I'll put in a small amount of detergent in this because what the detergent does is it decreases surface tension, so it decreases the propensity for this water to come back and stick to itself, right? And so I use common surfactants and then I put in cubosomes. So let's take a look at this a little more closely. So let's look at the first part, right? So this is 3 milliseconds, and 3 milliseconds is the time for this drop to come and go splat. Right? And in every single case, it doesn't matter whether I have cubosomes, if I have surfactant, if I have detergent, if I have water, it always spreads out to the same extent in every single case. What does that tell us? So you can go back, and one of the things that may be annoying for other people but really comforting for chemical engineers is to think about these in the context of what are called non-dimensional numbers, right? So you look at what are the things that control the physics in this problem, right? One of them is inertia, because you've got this drop that comes down and it's basically potential energy transforming into kinetic energy as it spreads. And this number called the Reynolds number tells you the ratio of inertia to viscosity. And what you see is this is a really large number. So inertia dominates this problem, right? You can also write down what's called a Weber number that, this is the density here. So it's inertia relative to the surface tension. And again, what you see is that the Weber number is huge, much larger than 1. And what that means is that this part only cares about the big mass of water that comes and hits the surface. It all spreads out in the same direction. What's more interesting is the second part of it, right? And this is the part that tells you about why the pesticide doesn't get stuck on the leaf, because if I have water, it just bounces right off, right? If I use detergents like these pluronics and tween molecules, then you can see that this retraction takes a much longer time. It's still on the order of 100 milliseconds, so it's not that much longer, but it's it's much, much, much longer than 20 milliseconds. And it doesn't retract to the same extent, which means that, yeah, surface tension, reducing the surface tension is helpful because the water then doesn't want to stick to itself. It doesn't mind being spread out a little bit on the surface. The contrast is with the cubosomes. 1% cubosomes, 1% of the detergent. What you see with these cubosomes is that it spreads out and doesn't move at all. Right? So there is absolutely no retraction in these systems, and this is qualitatively different. It's not just that you've reduced the surface tension. This is not a surface tension effect. This is something which is a completely different mechanism that you see in these systems. And so we went back and did ellipsometry and so on, and what we were able to show is that in that 3 millisecond time frame when the drop goes flat on the surface, the lipids that are packed into this nice cubosome FPN3M phase see the hydrophobic surface and realize that they would like to reorganize because, remember, they're held together by this very delicate balance. They can sort of flip around and form a very thin, 3 nanometer coating on the surface, and that's what does the trick in these, right? So in 3 milliseconds, it forms this very beautiful 3 nanometer coating on the surface, and that then helps you stick to the surface. So let's look at something more challenging. So, people who work in the area of wetting have this fascination with Lotus, right? Because if you look at the surface of a Lotus leaf, what you see is a hydrophobic surface with the same kind of microcrystalline wax coating. But in addition to this flat surface, there's all these nodules, right? So there's this range of roughnesses. There are micron scale roughnesses, there's nanometer scale roughnesses, and what that gives you is a surface that is super hydrophobic, right? Which means if I put a drop of water on the surface, it just slides right off. So Manoj went around all of Pune trying to look for Lotus. And if somebody tries to sell you Lotus, typically what they're selling you in Pune is water lily, which is not super hydrophobic. And so he found a place in Hadapsar which had a Lotus plant, brought it to NCL, and now there's a thriving Lotus garden in NCL as a consequence of this. So let's take a look at what happens when you take Lotus, right? So here's a drop of water on an inclined Lotus leaf. This is spectacular, right? I mean, look at this drop of water. It doesn't even change its shape. It just rolls right off. And that's typically what you'd see with Lotus leaf, right? That's the reason Lotus leaves are always so clean. Here's what you have with cubosomes. This is amongst the most demanding tests I think you can try. That's one drop there. In fact, you can load it up even more, put more weight on it, moves a little. You can just keep doing this, right? So again, what happens here is exactly the same thing. The lipid with this, which is the small molecular object, goes in, coats this stuff, and once you coat it, the surface properties are completely changed, right? And so we went ahead and did this on Lotus leaves, on a whole variety of things. You get this very beautiful coating, if you want to spray detergent, I think there's one of them. This is detergent, and after you spray detergent on this to try and help your pesticide to spread. Of course, this is just a detergent and no pesticide. One of the things you see is you get these dark spots. So if you look at the structure of the Lotus leaf, you see a lot of issues. So these are, because you have these detergents which are not really friendly, you end up damaging the structure of the Lotus leaf a lot. In contrast, the material that we started off with, the lipids, these are plant derived. So these are derived from sunflower oil, completely benign, non-toxic, food grade. You can eat this stuff and there's absolutely no damage at all to the Lotus leaf, right? So this is something that we were really fascinated by. Now, one of the things that people do when they spray agrochemicals is to worry about something called rainfastness, right? So we've had a tremendous downpour today, and one of the things you do when you just sprayed a bunch of agrochemicals is you don't want that kind of rain to happen because it just washes everything off. So rainfastness is something that's extremely important, and so we set out to try and check if our materials were rainfast, right? So we took some fluorescent particles. So these are fluorescent particles. Each particle is about a micron in size, so you can't really see individual particles. Took a commercial adjuvant. So this is what is used by farmers when they spray things. This is, you don't really know, it's some kind of a non-ionic called Lapsa 80. You can just buy it off the internet. And we mixed these fluorescent particles and sprayed these particles onto a hydrophobic surface. You take this, wash it once, every single particle comes off. It's just clean, right? On the other hand, if you take the cubosomes and you take the same fluorescent particles, first of all, you notice that the coverage, the way in which it spreads out, is a lot more uniform. and you can keep washing it. We've done up to 10, 15 washes, and it doesn't come off at all. So the rate at which it leaches off the surface is extremely small, even with very vigorous washing. And so this is something which is very useful for a variety of people in the agrochemical industry. And so I'm going to tell you in the last bit about some very recent stuff that we've been doing with this material. So there's a company, so I work with a large number of large multinational firms, but I'm really proud of this work. This is done with a company that's based out of Hyderabad. It's a relatively small company, and their key technology, their key intellectual property is a synthesis of pheromones, right? So pheromones are sex attractants, right? And this is what bugs, insects secrete when they want to mate. So one of the big problems when you have an infestation in a field is the extremely rapid rate, the exponential rate at which numbers of these pests grow. And that comes about because when you start off with a small number of insects, they when there's a lot of food, then they start mating, and then these numbers go up really exponentially in time. So one possibility is then to spray it with pesticide and kill these bugs, and sometimes they will develop resistance to these. Or what this company does is it has the ability to synthesize about 200 different pheromones that are specific to different bugs. And what these things do is it signals to a bug of the opposite gender to come and mate, right? Now, so this allows the other bug to geometrically, to sort of spatially localize to find out the other bug. Remember, you have a field and you've got a sparse number of these bugs. They really have to have signals, very powerful signals by which they can find each other to mate. So what these guys do is they spray pheromone everywhere. And what that does, it confuses the hell out of the bugs, right? So the bugs have no idea where to go, and they don't mate, and as a consequence, there's no infestation. These are natural materials sprayed in very small quantities because they're extremely powerful sex attractants, and so you don't need very large quantities of them, and therefore no pesticide, and at the same time, you get no infestation in these materials. So fascinating new technology. The problem with this is that these are extremely expensive compounds, and this company has a way of synthesizing many of these, but the problem is that you don't want to spray a lot of it because it's very expensive. It's not really an economically feasible solution. So one of the things they've been doing is they also work with the US FDA. They're one of their big programs. So it's a Hyderabad-based, it's an Indian company, but they've been looking at infestation in pine forests in the US, and they've run large field trials there. Especially to translate this technology to India, one of the key challenges is to use these expensive pheromones to make this affordable. So the solution they have is to take little plastic capsules, right? So they're like the balls that we showed, the fluorescent balls, but fill them up with a pheromone and they get them to stick to these plants. And that's where the technology that we've done is really exciting for them, because it combines their ability to make these pheromones with then the ability to try and get them to stick. And so that's something that we started off with, trying to understand how to get lipid assemblies to curve, and then along the way ended up discovering this really nice technology which is now being field tested, right? So that's all I wanted to say right now. So the idea is really, I think, the message I really wanted to convey is when you do the science, when you do the research, some of it is sometimes necessarily directed, especially if you work in industry. But if you work in national labs or in academia, I think the freedom to do interesting things, discover interesting things, can lead to technology. Not in every instance, but I think in sufficiently many instances that it's worth, I think, supporting this. You also end up discovering that not everything that you read in a textbook is necessarily the entire truth. Sure, I mean, something that goes into a textbook is reasonably field tested, but there's a lot more out there, and it's only by doing this kind of experimentation that you discover what's out there. So I think the most fun part of it for us is that we discovered these really nice predictive rules that tells you that if I have this kind of a molecule, it's going to give me an inverse phase, and that's something which is a very powerful rule to have. It has implications beyond just the monoline that we talked about. But along the way, we also came up with this really nice technology. And now this is undergoing field trials to look at a much more green-friendly way without using pesticides to come up with ways by which you can combat pests. So thank you for your attention, and thank you for coming despite the rain. Thank you.